Good morning, everybody. Good morning, everybody. Okay, so second day, I think it's going to be a wonderful day. We have a bit of a change to our program. So oh, JK will talk just after coffee this morning. I will fly through some of my slides. We'll make up some time there. And then Alejandro will start a bit earlier. And then we will move the coffee time a bit shorter as well, but it will move to a bit of an earlier slot. Okay, so that's the one. Then the second thing is I'm introducing Marcus Depfenart, and he can say a bit about himself, but he landed within this environment in quite a strange manner. So you need to say a bit about that. But I also want to thank him for all the artwork that you see, the banners and the logo and all of that was also his work. So he's not only a scientist and a surgeon, but also an artist. But he uses ChatGPT, of course. Yeah. Okay, Marcus.
It's not good that you told everyone. So good morning, everyone. Welcome to that second day. I must admit, I'm not a morning person, but I try to be awake. So yes, my background is actually a bit different to your background. I'm a plastic surgeon by training, but somehow I was pulled into peptides, into proteins, peptide design. I started off with peptide design, then protein design, then I ended up with nucleotide design, like aptamers. And it was a strange way, but somehow I got to vaccines. I don't want to tell the whole story how I ended into vaccines, but I'm often asked when I'm at conferences, at plastic surgery conferences, for example, or at advisory boards, what is the connection between this kind of work and plastic surgery? Because also I think for you it sounds a bit weird or strange. But then I always tell the people, you will remember me in 20 or 30 years from now. We will laugh about what we did to our patients when we did a facelift, because then we will say that was so cruel, like in the Middle Ages, and we will actually regenerate tissue rather than try to pull the face off and push it down back after cutting out a lot of skin. So I'm convinced that transient expression and gene therapies, regenerative medicine will be the future also in my field. And then I can say I was one of the pioneers in that field as well. So no, but so today I would like to talk to you about a toolbox, actually, because we heard interesting things yesterday, very interesting findings about in the neurological field, also with the trisomy, all very interesting measurements, epigenetic changes, but we have not heard yet much about tools, how to counteract, how to interact here, beside that super agonist of interleukin-15. So one of the toolboxes is that system here that I would like to introduce to you. So we are all very much aware of all kinds of viral systems, all those adenovirus that are used to transfect tissue for gene therapies, but also for transient expressions. But I think the whole trend in the market goes more into aviral systems. And aviral is very important because what we also saw from, especially when we looked at the vaccines in the COVID times, that the vaccines that used a virus as a vector, they were in some regions quite effective, in other regions not so effective. And I think it's not a surprise that AstraZeneca, for example, did not perform so well in Africa as it did, for example, in the US or in Europe, because it's a adenovirus from a chimpanzee, yeah. So and I think it's not a secret that the cross immunity is probably rather in an area where these animals are living than in an area where the animals are not based. So that's why, and those vectors, they can be just used once. So, and we also saw that in that vaccine that with the boosting, the effectivity was much lower than with the prime. And they actually tried to adjust then the doses a bit to have lower dose initially, to have not such a strong response against the vector itself as well. So we need those aviral systems. What I want to introduce to you is a transreplicon system for RNA production, but of course also for high yield protein expression. But before we go there, so now it's not moving. Okay. It's only moving on my screen here, but it's not moving here. You see, that's why I don't like mornings, huh? And the first is always the stupid one as well, yeah. So, okay, it's moving here, but it's not moving there. Okay, perfect. Thank you very much. So when I was asked to do a lecture here, a presentation, I was first thinking, what does that topic beyond the genome mean? For me personally, it means actually everything that is more or less against the central dogma of molecular biology, that it goes just one way, from DNA to RNA to proteins. And here we have different levels, I would say. The first level is epigenetics. We heard a lot about it. The next level is non-coding RNA, but also, and that's very important, untranslated coding RNA. We should not neglect that. And you will see that later. And you know of those quite well, I'm pretty sure, but I'm not so sure if everyone knows about post-translational modifications as a further level of beyond the genome. So the post-translational modifications have a large impact, not only on how a protein works or looks like, it has also a very, very important coding function. But we will also speak about that a bit later. So if we look into the literature, I was just scanning the literature a bit, the most recent literature you can see there's a lot about epigenetics for human diseases. There's a lot about epigenetics in case of non-coding RNA. We find interesting stuff about inhibitory RNA. We find small amplifying RNA. We find small activating RNA. So sometimes we speak about things like inhibitory RNA or the interference RNA, and we speak at the same time about pRNA, pVRNA. So it's all also from the nomenclature, it's very difficult because there's no real nomenclature out there. And if you look into the literature, you will recognize that actually the same thing is presented totally different with different names, and the same story is sold often to you just with a different name. So that's why it's a bit confusing, and it's really a thicket, but we see that this is really, that's the future actually. We are now beyond the genome. We are beyond encoding with DNA or with RNA. We now want to understand the other levels of transferring an information. And now I would like to introduce you to the that I called PC Origami 1.0. Sounds nice, huh? Looks a bit like the banners. No, but so this is a DNA-launched self-amplifying platform. It's a trans system. So why is it DNA-launched? So we could discuss that now for 2 hours probably. So I'm convinced that DNA is actually a better platform than mRNA, especially in our context here in Africa, because of we don't need that cold chain. We don't have those supply chain issues when we need nucleotides or so for manufacturing. but also we have a much smoother expression over time. So it starts very smooth and it becomes stronger and stronger and stronger with such a self-amplifying system. And that is actually very beneficial. We saw that from many people that got reactions. Now we can discuss, was it the LNPs or was it the RNA itself with the mRNA vaccines? But fact is that you actually exposing your cells to a large amount of foreign RNA immediately. And that is like an intracellular cytokine shock for the cells. And that's different if you use DNA. And of course, DNA is not so effective. That's a big downside. But one key to effort is the right administration. So traditionally, vaccines are applied intramuscular, like the mRNA vaccines as well. But intramuscular is not a good route, actually, because in the muscles, you don't have those immune cells. They have to migrate into the muscle first. So if you rather inject intradermally, for example, then you can actually transfect already immune cells, dendritic cells, for example. And that increases the efficiency dramatically. And there is one DNA vaccine currently in the market for humans. There are a few vaccines for animals, but for humans, there's one vaccine. It was launched in India, COVID-19 vaccine. And it did actually quite well with moderate doses, I must say. So I think now they apply 2 milligrams per shot intradermally. And they use a very old vector. The vector is from 1995. It's the PVAX1. It's the only FDA-approved vector so far. That's why they were using it, but it's actually not a very good expressing vector. It's a poor vector, yeah? And the results are quite good, I must say. So that's one thing, of course. Another thing is if you have a self-amplification on the RNA level, then you just DNA launch, and then you have the self-amplification on the mRNA level, but it increases over time. It's not immediately full power. It is like in a virus infection, and that is why it's also nice. I think always in vaccine designs, it's important to mimic the pathogen somehow in order to have an immune response that is more like a natural immune response. Now, the big downside or the big discussion about DNA vaccines is they might integrate. So, but that has never been seen in mammalians. But that's one of the points that always the opponents that are against DNA vaccines are saying. But that's why in such a system, it's important. This is a suicide system. That means all the cells that are transfected die within 3 to 5 days. They become apoptotic. So there's no chance of integration at all. That's also very important to know. So now let's have a look at it, how that works. So usually people are using those self-amplifying systems. They use alpha viruses for that. So there are old world alpha viruses and so-called new world alpha viruses. Old world alpha viruses like the Semliki forest virus seem to be somehow more effective. The reason for that is also what one could discuss over hours probably. But however, so we use the Semliki forest virus, and this is how it typically looks like. So it has those non-structural proteins. That's actually the replicase complex. It has a subgenomic promoter, and then it has the structural proteins like the capsid proteins, envelope proteins, and so on downstream. So now there was a group in Sweden in the 90s somehow, they started to use this and to make a vector out of it, a DNA vector. So they just took away everything that is downstream of the subgenomic promoter, and they insert their vaccine antigen there. And they had actually very nice results, and they're still very active with that at the Karolinska Institute. But as you know, before COVID, nucleotide-based vaccines were not really the choice, or had it very difficult from the regulatory side as well, especially in humans. So how it works is typically that one has this as a full system, and then sometime people went over actually to trans system. That means you divide the non-structural proteins from the structural proteins. You have the one construct here that is in form of an RNA, because people are also doing that with mRNA. There's now one mRNA vaccine has been launched in the US that takes this approach actually. But the trans system would mean you have the replicase complex in one construct, like one plasmid, for example, and you have the replicon. That means the part that gets amplified in another construct. So what would be the advantage of something like this? The advantage is that, of course, you just have to do your replicase complex 1. So you could, once you can mass manufacture it, that's very good. But you also can decrease the dose of the replicase in the cells. That's very important because the replicase is very toxic. And that's why you have an apoptosis after a few days, usually. And if you divide these two, then you can actually prolong the expression for a while. And another thing is, as you can see, this is quite large. This is already 8,000 base pairs. The replicase complex, you can imagine there's not much space for loading. So, and especially if you want to transfect cells, then you actually don't want to end up with too large constructs at the final end. Another thing, and this is actually quite new, what these people usually doing, you see those recognition sequences here. What we did and what others also now do is that we actually took that away from the replicase so that the replicase is not self-amplifying. Because if the replicase is self-amplifying, you end up with a large amount of replicase in the cell. You don't need so much replicase in the cell. The replicase is toxic, as I told you, and the replicase is actually occupying the ribosomes, and that decreases your yield actually. And I tried to illustrate the 2D structure of those recognition sequences. And that's very important. That's another thing in modern molecular biology. I think we have to look more visual into things. We have to see the things. We have to see how they fold. And that's another level of information, actually, how RNA is folding. Then here we have the two-dimensional structure that is needed for the replicase to recognize what the replicase will replicate, because all other proteins actually in the cell are shut down. And that's another reason for an apoptosis as well, because the cell is just dying from the metabolism goes down. And it's totally abused for the replication by the replicase. So this is how such a system would look like or looked like. So you have the replicase complex on one plasmid, and you have another plasmid that contains then your trans replicon. That means what you want actually to amplify. And here you see it again. That's for SARS-CoV-2, for RBD and nucleocapsid, the part. It's very important to also mention that we used the most tiniest portion that one can need as recognition. So there are other trans systems now. So BioNTech had the trans system. They have not used it yet, but they have published it sometime. And they use a large portion. They use, I think they use the full NSP4 or almost the full NSP4 as well. So because they did not really find the right recognition sequences, so they rather put everything in. With mRNA, it's also not so important. But I think whatever you don't need, you should not add it, because we don't know how that interferes in cells and so on. However, this is just a picture to compare the expression. So we have here an antigen expressed with a PVEX-Neo. That's the PVEX-1, but optimized to a high extent. So it's also very good expressing. And here we have the trans system with the replicase. You see, in the 100 times magnification, it makes a huge difference. We also transfect more cells with it, funny enough, although it's quite big. So, and this is with a larger magnification. And here we see nicer pictures after 48 hours. I forgot to mention that this was after 24 hours. And you see after 48 hours, the cells are very healthy. Funny enough, what we saw and what we recognized quite often is that the cells actually start grow and divide. So initially, this seems to stimulate the metabolism of the cells dramatically. then we see a very good antigen expression, and that's actually also key to effort here. You see all the red things, here's the DAPI staining of the nucleus, but you see the red staining, that is double-stranded RNA that we have stained with a special antibody. This is important to actually also see that the system is working, because if the replicase is working, you end up with a high amount of double-stranded RNA. That's the intermediate phase when the replicon, when it's copying the RNA, then you have always a complementary strand because it works in both directions. And that's why you have a lot of double-stranded RNA, and you see the whole cells are actually full with double-stranded RNA. And that actually also led us to the idea that's not only very good for vaccines, because you can imagine double-stranded RNA is a very good adjuvant, via toll-like receptors, but it's also a very good machinery to produce large amount of RNAs for non-coding applications or non-translated applications. So now let me quickly run you through our CEVAC tuberculosis study that we have done. So we used this vector system for a tuberculosis vaccine. So initially we started with 100 antigen constructs in silico, and we then nailed it down to 12 at the final end. So it was quite a bit, yeah. So everything was done in silico first. And so we ended up with 2 slightly modified vector systems and 2 different type of antigens. So we had an antigen called tuberculosis 1. Tuberculosis 1 was an antigen that contains 4 full proteins of tuberculosis and several epitopes of other proteins crafted in these 4 proteins, and crafted in a way as they are displayed in nature in their natural protein. So that's also a structural approach to see how does it fold in the real protein, and then you try to craft it in there to actually have it displayed like it would be in nature. Because just taking epitopes like short sequences doesn't give you the right tertiary structure, the right 3-dimensional structure, and that is a different antigen at the final end. So the idea was to craft it in there, and the TBC2 was a fallback strategy in case the TBC1 is not working, because we saw it in vitro, it was expressed very nicely, but you never know, in vivo it might be not so effective. Things are only partially predictive, but that makes it actually quite interesting to do such stuff. So we had then always one plasmid with a replicase, another one with the trans, with the tuberculosis, either TBC1 or TBC2 in 2 different vectors. And then we added SARS-CoV-2 as another one. So SARS-CoV-2 plus, in some case, part of a lysogenic MTB phage. So now you can ask, why SARS-CoV-2? No, we don't want to do another SARS-CoV-2 vaccine, to clearly state that. The idea behind that is a different one. The idea is that now I told you initially that we always try, or we want to mimic the pathogen in its natural surrounding. So MTB is an intracellular pathogen, so we don't secrete it. It stays in the cell, the antigen. So, but the SARS-CoV-2 antigen, it was designed that it gets secreted. So, and what we get from that is from the viral part here, increases the interferon response, of course, also via toll-like receptors. And we thought it might be maybe also a very nice adjuvant, keeping in mind that everyone knows now SARS-CoV-2. So everyone in the world knows SARS-CoV-2, has each body came across SARS-CoV-2 or was vaccinated. You might find some people somewhere in the rainforest or something like that have not heard, or where the body has not had interaction with SARS-CoV-2, but everyone else had interaction. So, and that's immune memory is actually very good, because there is a group in France, they use a tetanus epitope that they put to their vaccines based on the same idea that they say everyone is vaccinated against tetanus, so that if the immune system sees that, jumps on that epitope, then you make it, the whole antigen, much more immunogenic. That was the idea. And we also thought that if the one gets secreted and the other one stays in the cell, then the secretion make something like a trace towards the transfected cell and actually tracks the immune cells towards the transfected cell. And there is another thing that might be a bit, that is also now coming a lot, is that what was described before, that in Pseudomonas, for example, there was a group able to show that there is a phage, a lysogenic phage, so a phage that is not actually killing the bacteria, is working symbiotically with the Pseudomonas and actually hides it from the immune system. And how does that work? So actually for bacterial clearance, you need that NF-kappa beta that actually activates then to TNF. And if you have a viral infection at the same time, then what happens is that the viral infection via the interferon blocks the TNF secretion. And so you actually weaken your bacterial response because of the viral interaction. And that's very interesting. So this group actually vaccinated against that phage, and they were able to clear Pseudomonas, that is antibiotic resistance. So, and I think there are a lot of interactions like this. And and interestingly, we also see those lysogenic phages in MTB. So they are integrated in the genome of MTB. So there is an indication that the bacteria actually live symbiotically to some extent with some phages that only take over when the bacteria get very weak. And so we thought it might be a very good way to combine viral components and bacterial components to have a crossover as an adjuvant, but maybe also serving that mechanism. We haven't shown that mechanism ourselves yet, but we thought it might be beneficial. And that might, by the way, also be a reason we heard yesterday about the helminth infections and how that interferes with viral infections as well. So, so there might be a crossover between those immune strategies that are actually preventing each other. Sometimes it's beneficial, like in the Plasmodium maybe, sometimes it's not beneficial. So now for the studies, I quickly run through the studies. So what we did is we looked for safety, immunogenicity, effectivity. For immunogenicity, we looked into the humoral response, of course. I'm a big fan of humoral response. I think it's essential, and this was also shown nicely yesterday. If the immune response is broad towards Th2, that this seems to be beneficial even for things like malaria. And I think also for bacterial infections, it's not to be neglected, because when the bacteria, they have to come into the body first before they land intracellular. So, however, we looked into cellular response. For the effectivity, we did for SARS-CoV-2 a neutralization assay. And for the TB, we did a challenging study with the Kramnik mouse model. So first we started with rapid studies for immunogenicity. So why did we do that? Because we had so many candidates and we actually wanted to come to a short list. And we wanted to reduce our candidates dramatically, but it was not so easy because they all performed very similar in that study, to be honest. So they were all quite good. So we had a very good humoral response, as you can see here. And that's very interesting. On the right side, you see one of the antigens is the HSPX, that heat shock protein of tuberculosis. And you see at day 43, we get a much better immune response, humoral response against the HSPX than against the SARS-CoV-2. And if we just transfect with the SARS-CoV-2, then we have the same response here. But somehow the immune response is brought into the direction of the TB rather than to the viral response. And that was very interesting for us to see that. And we saw that all the time. We did many rabbit studies. We did many mouse studies with BALB/c mice just for immunogenicity. And, but in our case, it's actually beneficial because we don't want to make a SARS-CoV-2 vaccine. We are looking into a TB vaccine. So then we tested different doses and different candidates. And interestingly, even with 70 microgram, what is not enough, what a lot for rabbits, yeah, we got a nice response. And we think we can actually even go lower. And That's just because of the self-amplification of on an mRNA level. Actually, you just have to get 2 plasmids in one cell, and then it starts. And that's the difference to standard DNA vaccine, where you have to have much more plasmids in a cell. We did those neutralization assays. We saw a very good neutralization. This is here Omicron, but we did it against different variants. So the antigen for SARS-CoV-2 is also designed a bit different. So we have 3 versions of RBD that are predicted in a mosaic approach, and some portions of nucleocapsid as well. So now the interferon gamma response. So cellular immune response seems to be very important in tuberculosis, and you see we have a very good response. If we did a qPCR, we have a 224 times fold change in the response in comparison to the control, what is a significant cellular immune response. So we also looked into interleukin-17A. So I don't want to bore you with all the results. I just give you a selection, but the interleukin-17A is one of those big markers nowadays, and I think Ludina will agree to, because she's supporting many people with a lot of markers here, that this is one of the great markers that is currently described that gives an indication of protection and against tuberculosis. And that is actually also quite good. You see where we end up here, the control is always the blue one. So now quickly to the mouse studies. I mentioned shortly that we did a lot of immunogenicity studies. I don't know how many mice we have used up, to be honest, but we used a lot, and a lot of rabbits, and just for immunogenicity. And then, of course, the fillet of our studies is the Kramnik mouse study, where we had 114 mice included in that challenging study, BSL-3. And this is how it looked like. So we infected the animals. First, we vaccinated them, of course. So we vaccinated them. We did a prime, and we did 2 boosts. And some selected animals, we did even a 3rd boost. Why did we do 2 boosts? Usually, one does not want to do so many boosts. That's not applicable for humans. But there is that FDA +1 rule that says you have to inject 1 time more as you would do it later. I think it's a safety measure. Then in case there is some toxicity, that it will be shown then if you do a further injection. So then we did, in most of the animals, a first infection after day 42. In some animals, we did a second infection as well. So we did, the first infection was 34,000 CFUs, intranasal, what is a lot. And the second one was 55,000. And then we terminated animals from time to time to look into CFUs. And this is our survival curve. So you see the control group, and you see the other groups. And unfortunately, all groups survived so well, so that we still don't know which is the best candidate. No, we have an idea which is the best one because of the clinical scoring or the behavior, but we saw, and even the group K, and that's interesting, group K was just vaccinated once, just got a prime. It was not vaccinated more often, and it was also protected very nicely. And if we look into the average weight, so you see they are all still, they always gained weight, so they did not actually show large impact. So they had some small dimples in some groups when they got infected, but I think that's acceptable. And then we looked into the CFUs. For example, at day 20, we saw the CFUs are quite low, interestingly. Then it goes a bit up, and then it goes again down. And in some groups, we had, after 62 days, we had no CFUs anymore in the lungs to be detected. And that's a full clearance of the lung. And that's in that model, in that Kramnik mouse model, that is SST1 deficient, and that is so susceptible to tuberculosis. We have not found that in literature. So if we look into the histology, just very quickly here, so this is from literature. You see there, you're probably aware of that is those typical lesions, 1, 2, and 3. 1 and 2 are the, it's Irvine was doing that classification. That's actually quite nice in that model, that are close to the human granulomas, but have a different pathomechanism of course. And those lesions too are the lesions that typically lead to a very early lethal end of the animals, or that they reach their human endpoint within actually days. So depending on the dose that you actually apply. But you see, so these are typical lesions too, and we see here our lungs look actually quite good. And I don't want to stress that too much, but if one looks in literature, there is of course comparison with there's a quite recent publication where they used the attenuated MTB and H37RV, and then they vaccinated the animals with that, and afterwards they then infected the animals with H37RV. What is also a bit an issue, I think, always, because we see that with those influenza studies as well. If you choose a strain to infect, and if you use an attenuated strain to vaccinate, then it's a bit like a cheat for me, because in real life you cannot choose the strain that you meet. That's the problem. And so that's why the protection against the clinical strain would be much more interesting. But you see the survival curve is pretty much the same, but the CFUs are much more. And if we would compare our one to that, then just the moderate would be similar to there, and moderate was known. We have one that was marked, and it was the group K, so that would be something like this here. So now very quickly, because I know I'm consuming up a lot of time here, I would like to introduce you quickly to the so-called origami code. And I think we discussed up and down what is the reason why this was so effective. And one of the reasons is probably that we are convinced that we are able to at least partially circumvent the post-translational modifications of the mammalian cells. And why is that important? I think it's important because the mammalian cells are not producing, and that's a big disadvantage if you use nucleotide-based vaccines. Nucleotide-based vaccines actually always lead to a mammalian protein. So, but if we are dealing with, not with viruses, viruses abuse the replication machine or the cellular machinery for their own replication. But bacteria or other pathogens like fungi or plasmodium or whatever, they don't use that. They have their own metabolism. And that's a disadvantage, because we then have actually different proteins. Viral proteins are mammalian proteins, but bacterial proteins are bacterial proteins. And I think that's really important. And nucleotide-based vaccines always end up with mammalian proteins and not with bacterial proteins, not with other pathogen proteins. And what we did is actually we circumvented, because one of the biggest difference in those proteins is the post-translational modification. And it's important to display the right antigen towards the immune system, because if you display the Achilles heel at the neck, for example, then the immune system has a different target as if the Achilles heel is really at the Achilles heel. And that might make huge differences in nature, and that might be a reason why nucleotide-based vaccines are not so super well performing against bacterial targets currently. Okay, I'm running out of time. Yeah, I'll quickly show you. So we predicted the post-translation modifications, and then we did a study with GenScript where they were actually looking into the post-translation modifications. So we expressed the same antigen in a standard vector, DNA vector, then in the origami system, and then we expressed it in E. coli. And the result was there was no difference when we used the origami system to the E. coli protein, but there was a difference when the standard vector was used. So I'm out of time, or how long do we have? 2 minutes, huh? Okay, so another thing that we did is, that's a different story, but we then thought that we can actually just use maybe nucleotides and work on a different level. So untranslated RNA and we know that the system produces masses of RNA, and we thought maybe we can use this also to train immunity just with RNA. Let's jump over this. You all know that. So there is in nature there is that RNAi system that, for example, small animals like mosquitoes are using, or Drosophila, because they don't have a real adaptive immune system. They have no humoral immune system. So they actually get in touch with the virus, and then they dice it in portions and send it around with those called hemocytes. Those are immune cells, and so they spread the information. And epigenetically, they then are able to control those infections just by producing complementary RNA. So, and there's just a brand new publication where they did something similar. And what we did is actually, so important, maybe just important is that you, of course, you have to balance the amount of RNA plus the interferon response, because the interferon response will actually block you. That's the disadvantage mammalian. You have to choose the right sequences. You have to take care that the RNA is cleaved correctly. And you have, of course, to spread the information, and you have to get an epigenetic imprint somehow. So we did a challenging study with Charles River. So we have chosen portions of the antigenes of influenza, but without start codons. Actually, we included several lines of stop codons before that, and those are all conserved portions. We put it into the vector system, and this is, you see here, you see again a lot of RNA, double-stranded RNA. And at the final end, just to see, this is our survival curve. So we have a bit more than 50%. So it's not perfect yet, but you see, this already gives you protection. And the animals were actually challenged with the Puerto Rico strain, and the construct was not made specifically for the Puerto Rico strain. It was a consent sequence. So, and you see that other vaccines that are, for example, when the animals were vaccinated with Puerto Rico strain and were infected with the California strain, that they only had a protection of about 20%. So our 50% are not too bad at all. So, and this is also the clinical score where you see this is also not too bad at all. So, okay, that's so far everything. Sorry for my overtime, but I just wanted to introduce you into the platform and potential applications, because I think this goes far beyond protein synthesis and beyond the genome. Thank you very much.
The question going to the tuberculosis data. So the outcome seems very, very interesting. So do you know the immune answers? So you haven't specified anything as development of B and T cell answer. I think the previous success on the tuberculosis vaccine was based on T cells. So how is it in this case?
Thank you very much for that question. So I think it's actually a combination. You're right. So, and I think I tried to touch that initially, that I'm not completely convinced that only T cells make it. I think it has to be a combination, and there is a reason why we have those several lines of defense, and we should actually use them all if possible. That means we should use the innate, but we also should use the adaptive, and that means the humoral and the T cell response. We have a T cell response, as you saw. That was very substantial, but we also have an antibody response. And you're right, usually the studies don't look in the antibody response at all. So we hardly find studies where they actually show the antibodies. The question is now, don't they have antibodies, or are they just neglecting it? I don't know. Fact is that those bacterial antigens usually are not so good for a humoral response in general. And that might be a reason why they have so poor antibodies that they don't want to show them. But I don't know. But we are convinced that both is essential. And you're right. And initially also, in order to select, we hope that we have a humoral response, because it's very difficult in the rabbits. The rabbits are easy because you can draw a lot of blood all the time. So that means you can do ELISAs, and you're not limited like with the mouse studies where they, you know that better than me, it's very difficult to get enough blood to do a lot of ELISAs. So, but doing T cell response is quite an effort. If you keep the animal alive, it's an effort. And if we then have to kill the animals all the time, sacrifice them before the time, then it's also costly, and the preparation from rabbit is also costly in that case. So that's why we hope to see the humoral response as a selection for immunogenicity, but we were actually quite surprised that we had such a good humoral response. And we think it might be both, but it's actually, you're right, people are thinking that the cellular response is more important, but that was also good. Yes, you actually, you need the memory. Yeah, that's important. You need a long-term memory. That's right. Yeah.
I think we've got, is Alejandro